Our guest today is Dr. Ewoud Schmidt, who is an assistant professor at the Department of Neuroscience at the Medical University of South Carolina. His lab investigates human-specific genes in brain development, function, and disease. We're really excited to have an interesting discussion with you. Thank you so much for joining us on the podcast today.
Thanks for having me.
So before we really dive into your research on human-specific genes, I thought we could sort of start with the bigger question of, human evolution. So what are some ways in which humans actually are behaviorally specialized compared to some other species?
I mean, if you were to try to define what makes humans unique or specialized, unique is a loaded word, I think, but it's our cognitive abilities. And that shows up in all sorts of things that we do. I mean, the fact that we do a podcast here, right? It's like no other species would do that. The technologies that we develop. So with that comes a whole cultural evolution. So I think we could, our cognitive and learning abilities seem to be, in a way, foundational to that. Because anatomically or physically, we're not the fastest runners, we're not the best climbers. It's really this cognitive capacity that seems to set us apart. Yeah.
So what are some of, like, if you think about the broad scale of evolution of mammalian species, like in general, and maybe specifically to humans, what are like some of the main factors that drive that kind of speciation and like specialization of different types of species, or even at the level of brains? Like, what are the main components intrinsic to us that allow those different types of specializations to emerge?
You mean from a biological perspective? I mean, the way we think about it is that there must be unique genomic changes that underlie our brain evolution. And when we say unique, it's sort of that's a long process. Different changes that appear in different species that eventually, lead to human evolution. And then when you look back now, you see certain features like large segmental duplications and specific genomic variations that are unique to humans that we tend to look at least in the context of human brain evolution, that we see in humans that are not necessarily present in other species. That's not to say that we should only focus on those. There's a lot of interest also in primate-specific changes in the genome, because we shouldn't be too focused on just humans to understand human evolution. We are a branch on this large evolutionary tree, but that's where we tend to look to understand what makes humans unique. We start there, and then from that, genes encode proteins, and there's a whole molecular and cellular pathways where we try to figure out how they are different in humans, and then ultimately how that relates to brain function and behavior.
Yeah, and I think, one thing that's really interesting about your research is like the types of findings you can have by studying human genes in the context of brain development, brain organization, and even behavior are applicable to brains in general. These are sort of trying to, it seems to me, like unravel some of the basic rules by which, like, those types of processes and structures are regulated. Getting back to evolution a little bit, if you compare different, even just within mammals, say humans and mice, what are some of the sort of divergent physiological or anatomical properties that make humans special? I mean, presumably there's differences in brain size, things like that. But if you compare even at the level of microanatomy, what are some fundamental evolutionary changes that humans have adopted that maybe, or primates have adopted maybe, that rodents haven't?
Well, in terms of anatomy, I mean, if you put a mouse next together to a human, they're very different anatomically speaking. And I think it's a good question to ask because, we tend to be very much focused on what makes the human brain unique. But the human brain doesn't evolve in a vacuum. It evolves in response to changes in our anatomical our anatomical makeup, the fact that we are bipedal. So there's all sorts of characteristics about what makes a human anatomically unique that, of course, relates also to how the brain has evolved. And there is an interaction between the two. In terms of what makes humans anatomically unique compared to rodents, I mean, that's a long list. And which ones have really contributed to the emergence of our brains and cognition? That's, of course, a sort of an open question there.
So what about our brain specifically, since we are neuroscience?
Yeah.
Because, of course, we always jump right to the brain. Are there known sort of structural or functional changes that we see that are unique to human brains as compared to some of our close ancestors?
Right. So that's been studied for a little longer, and like definitely in the past several decades, we've learned a lot more about some of the things. Like we have to be careful calling them unique features or specialized features about what sets the human brain apart from maybe other primates or other mammalian species. I mean, the first thing that comes to mind, and I think rightly so, is brain size. We have a big primate brain, but what we've learned also from many studies in the past is that there's a change in the way neurons are connected with each other, and with the idea that they're more densely interconnected, that connectivity, especially in the neocortex, has increased, and that's thought to underlie some of our cognitive abilities. So neuronal connectivity and number of neurons is one of the first things we tend to think about when we say human brains are unique. Or have specialized features. I'm sure that's not the full list. There's definitely physiological properties, and as we hopefully study them more and do more comparisons between different species, I'm sure we'll uncover more sort of like, I would say, specialized features that would characterize the human brain over other primates or other mammalian species. And that's also where we start to need to better understand what makes it, for example, a primate-specific effect, or what is really human about that, or what is more mammalian-like, to really map that out. But when it comes to the question you asked, I would say neuron numbers and connectivity is one of the first things we like to think about and what we like to study actually in the lab.
Yeah, and you've been focused on looking at some of these changes that might be related to human-specific genes. And I wanted to just know, how many human-specific genes are there?
Well, it depends a little on how you define them. If you talk about human-specific gene duplications, I think we tend to number them in terms of like about 30. Depends a little on how you define that, how strict your definitions are. You maybe can make the list a little longer, up to 60. But there's a ton more variations. Human-specific gene duplication and human-specific genes is just one aspect. There's lots more variations that drive changes in gene expression, that there's a lot of studies now that really start to map what these are. And that's a long list. And we're now seeing not just duplications, we're also seeing human-specific deletions. So I would say it's an active field of research. I'm sure that list will still grow. And as Mart says, we tend to say like we share a genome of 98% with chimp, our closest relative. There's a lot that can happen in that 2%, if that is even still accurate by the time we're really mapped these.
Interesting. And so your work has mostly focused on one particular human-specific genome called SR-GAP2C.
That's right.
And really looking at its function sort of across many levels. But let's start sort of at the molecular level. What is this gene doing, do you think?
Right. So SR-GAP2C is a human-specific gene duplication that was duplicated from what we call the ancestral copy SR-GAP2A. And SR-GAP2A is present in rodents, in many other mammals. And this was actually discovered before I started working on that. This was done in Frank Pelleut's lab, where they discovered that SR-GAP2A, the ancestral copy, plays an important role in regulating how synapses are developing. And it actually plays a role in regulating the maturation of synapses, of both excitatory and inhibitory synapses, and it limits the number of synapses that are formed. And what they found, and this was also in Frank Pelleut's lab and by Cécile Charrier, she did amazing work discovering that SR-GAP2C actually inhibits all those functions. So on a molecular level, it inhibits the function of SR-GAP2A, and then we uncovered that this may happen actually on a protein level. The two proteins bind together, And when they bind together, it leads to a degradation of the proteins, effectively lowering the ancestral copy. And when you inhibit the ancestral copy, you get basically the opposite effect. Instead of promoting maturation, you now slow down synaptic maturation. And instead of limiting the number of synapses, we're now seeing more synapses. So that's
like kind of insane to me that part of human brain evolution was to duplicate a gene that served the function of regulating dendritic spine stuff, and the duplicate inhibits the existing pathway. So the evolution is basically that you have a new protein that degrades a pathway for regulating spine growth, spine development.
Right.
And now you have these extremely spiny, synapses,
right.
or spiny dendrites. That's crazy.
Yeah, and in a way, you could wonder why not just delete the gene.
Right.
And when we talk about evolution, it's hard to, like evolution, there's no
rhyme or
there's
reason.
Yeah, right. It may have just happened like this. Or you could also argue that there is a difference between deleting a gene and having a new gene inhibiting the original, because that gives you a level of regulation. You can
flexibility.
Right. So we don't know if that's the case, but there may be more to it, to that mechanism that we haven't discovered yet. But they're not the same thing. Removing a gene or duplicating a gene that then inhibits the ancestral copy is a different mechanism.
Yeah. So I guess the long story short then is that if you were to take this human-specific gene SRGAP2C and express it in a brain that it doesn't normally express in, you could ask questions about its functional role in regulating many different things. So what is your approach for taking this gene and then trying to understand at various levels of brain complexity or brain organization the impact of that gene?
Right. So one nice thing about this mechanism is that we can model it in mouse, because mouse has SRGAP2A. So by putting SRGAP2C in there, it acts through that same pathway. So what we can do is we can take that human gene, express it in mouse cortical pyramidal neurons, and then we have different tools in our toolbox, if you will, that allow us to, for example, map if a neuron has all these extra synapses, all these extra possibilities to connect, how does that affect the overall connectivity of these neurons, basically the inputs that these neurons receive? And there are some very powerful tools these days. One of them is called monosynaptic rabies tracing. And the interesting thing about rabies is that rabies as a virus is so lethal because it's very effective in jumping from one neuron to another. And now we can actually use that virus to map how neurons are connected with each other. So by using those kind of tools, we can map how connectivity changes for a neuron that expresses SRGAP2C. So that's we call it the structural connectivity that we map. And then there are very powerful these days imaging technologies that allow you to also look at neurons in action as a mouse, for example, behaves in a certain paradigm. So then we can ask, okay, if neurons express SRGAP2C, what are they functionally doing under certain conditions? So we can map that and then figure out, is that different when they express SRGAP2C? So then ultimately, we relate that into behavior, where we can also ask mice to perform certain tasks, test their performance, and see if the human gene, the expression of a human gene, impacts their behavioral performance. So those are all sorts of tools we can use to map structure, function, and behavior, almost sometimes it sounds like the holy trinity in neuroscience, right? It's like the way we study the brain.
Right.
Yeah, so that was a really beautiful explanation. It sort of gets at the power of the approach. So maybe we can just talk about some of the actual findings that you had. So let's start sort of on the structural and connectivity level. What did you actually find?
Yeah, so when we express SRGAP2C, and we do this in specific cortical neurons, but when we map their connectivity, these neurons on the normal in a normal wild type mouse, they receive inputs from other cortical regions. We call this cortical connectivity, and they get subcortical inputs, and those are functionally different. And what we see is that when you express SRGAP2C, it increases these cortical connections. But we didn't see these inputs from those subcortical regions to be increased. And we got really fascinated by that, because one of the features that are interesting about the human brain is increased cortical-cortical connectivity. Now, I don't want to suggest that that's what we're actually modeling. Why humans have more cortical connectivity is really because of SRGAP2C, but there seem to be some interesting correlates there. So from a structural point of view, we increase selectively those connections when we express SRGAP2C.
Yeah, and it's really powerful that you can see sort of selective changes in certain circuits as opposed to others. Do you have any idea where that specificity is coming from in terms of mechanism?
Right. So that is a question we're very interested in. And one of the things that we noticed, although I don't think it's a full explanation, but it's interesting that SRGAP2C, when expressed in the cortical neuron, it doesn't just upregulate the number of synapses equally across the neuron. And there seems to be some selectivity on where the synapses are increased. And we know from these cortical pyramidal neurons is the inputs they receive are in a way segregated and domain specific. So that could be one explanation. By targeting specific synapses, SRGAP2C has a specific effect on the connections that are being formed. That's one model. We also know that these synapses mature slower. And if you change maturation of these synapses, developmental timing is everything. When the brain develops, everything runs on a clock. Now we're slowing down synaptic maturation, and maybe that's causing some of these connections to wire up differently. So these are hypotheses that we have that we're hopefully be able to answer at some point. But that may explain to some extent how SRGAP2C has selectively targets those connections.
So then to sort of follow up and ask like the next question that you outlined, like what's the functional consequence of maybe some of these circuit level differences? You chose a really interesting set of experiments using the mouse barrel cortex, which is a very specialized structure for rodents that represents the individual whiskers of a rodent. Why that system, and what did that allow you to ask about the functional properties of the neurons that this mutant mouse had that a wild type didn't have?
Right. So it's a bit of it's often when we do experiments, it's a combination of just practical.
Mm-hmm.
We find it's the experiments that we use or the ways we target these neurons. We use specific approaches. The barrel cortex is very easy to reach. So that's a very practical reason. What is interesting about the barrel cortex, though, is that the circuitry is very well worked out. So we know very well how neurons are connected in that region. And for a mouse, the barrel cortex and which processes whisker information, the whiskers are incredibly important for mice.
Mm-hmm.
That you could almost argue they use their whiskers the way we use our hands and fingers.
Mm-hmm.
So it's a very important region for the mouse, and that allows us to test the function of neurons, and also related to behavioral paradigms that we can control very well. So, the combination of practical factors and the fact that we know the system very well gave us a good model to test what SRGAP2C does on a functional level. Yeah.
So, then you can set the mouse up with an experiment where you're, like as you said, using 2-photon imaging to visualize the activity of these cells while the whiskers are sensing some kind of tactile stimulus. It's a very, pretty, I don't want to say simple experiment, but it's a very straightforward experiment where you can present a stimulus, remove a stimulus. What did that allow you to tease apart in terms of the functionality of this gene with respect to like excitability of neurons?
Right. And there it comes in that the system has been described very well. And you're right. It's, like we're so used to these techniques that for us, this is sort of a most simple experiment you can do. It's also a system where the mouse doesn't have to learn anything. It's just a passive stimulation. But one thing that's nice about it is that we know what kind of responses we should expect. When you give a whisker stimulus, you expect certain types of responses in these neurons that we're looking at. So that gives us a good reference for what happens when we express SRGAP2C. And one of the things we saw was that this is a system that these neurons, they tend to actually, you could almost say they don't really like to respond to a whisker stimulus. We call it the probability of these neurons to respond to a whisker stimulus is very low. And we saw the probability go up dramatically when we express SRGAP2C, but very specific to when we applied the stimulus. So these evoked responses were the probability went up. And then overall, their spontaneous activity was much lower. So the evoked over spontaneous ratio, if you will, was increased when these neurons express SRGAP2C. And there's a way to look at that, from a sort of a signal to noise ratio perspective. And you could, if you then look at these neuronal activity patterns, it seems like the signal to noise ratio for the whisker stimulus goes up. And that makes it easier to decode when the whisker stimulus is happening from a population of neurons that express SRGAP2C when we compared it to a wild type mouse. So in that sense, the neuronal responses were changed in that they, you could say in a way, more reliably responded to the whisker stimulus.
Yeah, I mean, is it fair to say that they are more information dense? Like they basically provide more information to the animal by amplifying the signal and reducing the noise? Like,
yeah, right.
Real information.
That's how we like to talk about it, and I think that is a way to. For us, it's a starting point to think about how these neurons change their activity patterns. If we really want to start talking about information, I think it's then we need to start doing experiments where the mice are also doing a behavioral task and we know what kind of information they're processing. Because for the mouse in this particular case, the whisker stimulus is not very informative. It's if anything, maybe somewhat annoying for them. So if we talk about information processing, we may have to push this beyond what we did there. But it's a way to how we think about how these neurons have changed in their activity patterns.
Yeah. So you saw these, changes in sort of connectivity and then the functional properties of how the neurons responded to sensory stimulation. But ultimately, you want to try to put these together and try to understand how, the addition of this gene affects animal behavior. So what sort of approach did you use to begin to address that type of question?
Right. So we decided to stay again in the barrel cortex and test something that the mice need to use their whiskers for. And a very well-established behavioral paradigm for that is called texture discrimination. And it's a rather simple idea where the mice in the dark, so they can only use their whiskers, they detect textures that are coming in, and they respond. If there's a smooth texture, they get a reward on the right side, for example, and if there's a rough texture, they get a reward on the left side. Mice are pretty good at doing that. And what we decided to do was make that task more difficult by making the textures basically more similar. So for them, sitting in the dark, it becomes more difficult to distinguish between those. And then what we observed was that we made it actually so difficult that about 40% of the animals, the wild type animals, they were just not able to learn this task. So if you then look at their performance, they're basically 50%. They're guessing. This is just, I think it's left or right, I'm not sure. But when the mice express SRGAP2C, about 90% of the mice could actually learn how to do this task. Wow. So we saw an improvement in sensory motor learning, if you will.
Yeah. So this raises a lot of questions. So first of all, how specialized are these sort of behavioral kind of enhancements that these mice receive? Like how restricted to the domain of, perception or sensory detection are these changes? How do they translate into the other behaviors that the mouse does for mouse reasons, for non sort of operant conditioning reasons? It sort of raises a lot of questions about what has happened to the whole brain rather than simply, the apparatus for detecting whisker stimulation. Are there any hints about how this has improved memory or cognitive abilities in general, or even just sort of ethologically relevant behaviors in the animal? Right.
So that's kind of the point where we are at now. Those are all very good questions, and the simple answer we don't know, but we're actually testing that. So. And we're actually using the same sort of paradigm of texture discrimination, but we can modify it in such a way where we can ask questions about memory, for example, or we can test for these animals. I mentioned that we make the texture more similar. We can basically test them on different textures that are more similar or less similar, and we can see how does their performance go down basically as the textures become more similar. So that's more sensory processing. So we have all sorts of ways of changing the task where we can start asking those questions. And we have some interesting data, but everything is still very preliminary. But hopefully we'll be able to tease that out. And I think there's another sort of interesting question in there, which is, Is it true for maybe visual processing, or is it true for social interactions, things that we're not necessarily testing right now? And some of these we don't think they're involved, like social interactions, maybe not, but we haven't tested this in other sensory domains even.
Sure.
So yeah, there's a lot more work to be done to see how much this sort of generalizes to other behaviors.
I think one thing that your answer points to a little bit that's really powerful about this approach in general is that it's building off of known paradigms for testing out the functional roles of specific circuits in the mouse. So, hippocampal prefrontal cortical circuits have specific functions. There's circuits for emotional regulation. There's circuits for contextual fear learning, things like that. It'd be interesting to see, does everything just get enhanced across the board? Are there specialized circuits that take advantage of this, this change in gene expression? And that leads to one other question I have, which is, this is one gene, right? And you said, there might be lots of these types of human-specific genes. What do you think the collective impact of all those different sort of human-specific genes, like if you could do a crazy experiment where a mouse expressed every human-specific gene, I don't know if it's possible.
Right.
Seems tricky. What level? I mean, you've already demonstrated really robust impact on brain circuits in general with just one gene. What is your sort of thinking of like how all these other genes would factor into that?
Yeah, I mean, it's hard to predict. And even if you were to express all the human-specific genes, you're not generating a human brain. Like, you're just still doing that within the mouse context. So I think what we're learning by, for example, looking at what is specialized, unique about the human brain is that there's all sorts of aspects about brain development and brain function that you can change and that can have an impact on function and behavior. Think about plasticity mechanisms, think about connectivity, neuron number itself, just the sheer number of neurons, how those neurons develop, the developmental time points that they reach. And so I think all these different human genetic modifiers probably all play different roles in modifying these different processes. And together, they lead to something that what we then know as the human brain, but that still happens within a larger context of the human genome. Right.
I mean, like, I guess one related question is, what's really cool about this is you could even isolate specific human-specific genes and test their functional role the way you've done with SRGAP2C. Do you think there's any possibility that SRGAP2C expressed in the context of another one of these genes might alter the functionality of it?
Yeah, so that's the difficulty in terms of studying human-specific genes. And actually, in a way, it's the difficulty in studying any gene, which is if you knock out a gene, for example, the effects, how much are they dependent on other factors? And you're totally right. Human-specific genes may act differently depending on other players that are around. I mean, the interesting thing about SRGAP2C, and I think we got, I wouldn't say lucky with that, but it's a very interesting model to test this, is because it functions through a pathway that is present in mouse. And we also know, actually, if you knock out the ancestral gene, a lot of the phenotypes we're looking at are reproduced that way. So it doesn't mean that SRGAP2C doesn't have any other functions through other interactions with other proteins that are either present in the mouse or that you would need in humans. We would have to map that out. But I think a lot of the things that we're studying now work because of that pathway being existing in the mouse. But we're very much at the beginning of this. Like, the idea that we're studying the functional impact of human-specific genes on mammalian brain function is, I mean, I find it personally wild that we're working on it now. But we're very much on the early stages. There's a huge amount of work.
There's so many questions.
Exactly.
Yeah. It's a good spot to be in for an early stage.
Right. And there's so many new tools being developed. I mean, you must have heard of organoids, xenotransplantation studies where we preserve, human neurons. So there is also, as tools are being developed, we'll be able to answer those questions better. So I'm sure we'll be working on this for a long time to come. Yeah.
Very cool.
Yeah, it's really exciting. And I like that you brought up the topic of organoids, because one thing that I was really excited about, the possibility of using these sorts of approaches is sort of the benefits of studying human-specific genes and relevant to how that allows you to study neurodevelopmental disorders. And I'm wondering if you could just sort of expand on the power of this sort of approach.
Right. So to one extent, so there's a question I think that so far we haven't really been able to explore yet, which is do human-specific genes as genes that regulate synaptic development, for example, do they modify the phenotypic expression of neurodevelopmental diseases? A lot of neurodevelopmental diseases that until now we've been, for example, studying using mouse models, we knock out a gene that we know is involved or mutated in a certain developmental disorder, but we knock that out in the context of a mouse neuron. The question there is, does the presence of SRGAP2C, would that modify that phenotypic expression? So we can model that in mice, but there's a limit to that, of course. And other tools like organoids are very interesting because you can study some of these things within the context of human biology, human neurons. And I think at some point, and as some people have started doing, you can bring some of these models together. You can transplant organoids into mice to some extent. So I think there is a lot of work to be done there, and how much human-specific genes either themselves are playing a role in neurodevelopmental disease or are modifiers of the phenotypes that we're seeing in neurodevelopmental disease. That's something we have to work out. We haven't even really begun working on that question just yet. Yeah.
So lots of questions, lots of areas to explore for many, many years. One thing we like to ask on this podcast is, are there any projects going on right now that you see getting off the ground or in the sort of near future where you would be hiring, say, grad students or postdocs where, we have a lot of them listening to this episode right now. Are you hiring right now? Are you looking for people to help with these types of questions?
We definitely have some projects that we could use people for. And one of the questions that we're really interested in is we've been very specific in, for example, testing SRGAP2C in a specific cell type under specific behavioral conditions. And some of the questions you asked were very much about, okay, so what are the broader implications? What about other cell types? And there's still a lot of work we need to do there. So if people are interested, then they should definitely reach out because I think we have some very exciting projects going on that definitely need people. Yes.
Cool. Well, Dr. Schmidt, thank you so much for joining us on the podcast today. Really amazing work, and best of luck with all that you're doing.
Thank you so much. Thank you for having me.
Take care.
Thanks for watching today.
If you like this video podcast, make sure to subscribe to our YouTube channel for more cool neuroscience.